1953 saw the publication of two momentous scientific reports, which thereafter have received very wide acceptance, but ironically, much different in effect. In 1953, James Watson and Francis Crick published their article about the double helix model for DNA. Of course, DNA is the key to life's diversity. Hereditary information is transmitted across generations by means of a code that's resident in the specific sequence of certain constituents of the DNA molecule. In the same year, Stanley Miller offered experimental support for the first step toward confirming the theory of prebiotic evolution. See, if you're an evolutionist, you have to hold not only that the liver fluke develops gradually and so forth, that the eye and the lung and the heart and so forth all develop gradually. Take care of all these other problems, you also have to explain where life came from. And the answer is prebiotic, pre-life evolution, the chemical stage of evolution. And Miller offered what he thought was experimental support for the first step toward confirming prebiotic evolution. Both of these in 1953. One was a discovery about life's structure. The other was a discovery allegedly about life's origin. Little did the scholarly world at that point stop to recognize or appreciate the tension that was implicit in the two new insights. Because any secular theory of evolution requires an account of prebiotic evolution, where chemical evolution causes life to originate without supernatural intervention. There must be a preamble to biological evolution. It's plausible. Is it plausible that unassisted matter and energy organize themselves into living systems? Is that plausible? Now, Stanley Miller, using an electric discharge apparatus, identified several racemic protein-forming amino acids. Subsequent simulation paradigm experiments for an environment that was thought to be similar to the primitive Earth's precursor to the first living cells yielded some detectable amounts of most of the major kinds of biochemical substances. Presto, it seems like we're on the verge of being able to develop a prebiotic theory of evolution. But since 1953, most all of the hope of doing that has been really deflated. Current laboratory and theoretical research has reached an absolute impasse on this matter, and that according to Dean Kenyon, who in 1969 wrote the book Biochemical Predestination. He said it's not going to work. There's an enormous gap between the simplest living cells, which by themselves have an amazing structural biochemical complexity, and even the most complex protocell model systems which have been produced in laboratories. Moreover, he points out, what's the pathway for prebiotic synthesis of the relevant organic substances? Moreover, the experimental conditions in the studies of prebiotic evolution have been so artificially simplified as to be irrelevant to the processes on the primitive Earth. I have other details here about polypeptides and the presence of oxygen and so forth. The assumption that the early Earth was anoxic, without oxygen, has been pretty much discredited. And so if you have to have an oxygen-less environment for these chemicals to get together and do this, the fact that there was oxygen on the early Earth, from all that we can tell, indicates there couldn't have been prebiotic evolution. He points out there's no explanation even remotely possible or plausible, even after prodigious years of effort, for the optical isomer preferences that are found universally in living matter. That is, an isomer is two things that have the same number of atoms of same elements, but in a different structural arrangement. And in all living matter, there's an isomer preference for L amino acids rather than D amino proteins or D sugars. What accounts for that if it's all by chance? Why is it that all living things, we have the same number of molecules and so forth. It's an isomer, but it's always of the L, not the D version. And what's the origin of genetic, biologically relevant information in biopolymers, he asked. No experimental system yet devised gives the slightest clue as to how these biologically meaningful sequences of subunits might have come about in prebiotic polynucleotides or polypeptides. So what we're talking about is these sequencing, minimal sequencing units in DNA. How did that come about? And if you don't have the sequence, if you don't have information, as it were, stamped into the DNA of that form, it won't work for replication. So the scientists are just, this got to blow your mind. Charles Thaxton, Walter Bradley, and Roger Olson, in the book The Mystery of Life's Origin, published in 1984, pointed out that based on geochemical assessments, both the atmosphere and the various water basins of the primitive Earth contain destructive interactions which have vastly diminished, if not altogether consumed, all the essential precursor chemicals. Moreover, the soup would have been too diluted for direct polymerization to have occurred. Moreover, no geologic evidence indicates that any organic soup, even the smallest organic pond, ever existed on this planet. They point out faulty assumptions about the anoxic condition of the early Earth atmosphere. They point out that the experiments that have been done have got to be disqualified by illegitimate interference, that is, information interference from the investigator in the laboratory situation. They say the notion that the Earth was an open system with energy and mass flow to take care of the second law of thermodynamics still cannot adequately account for the configurational entropy work necessary for coding the early chemicals so that it would be valuable for the reproduction of life. Well, on and on and on it goes. And I'm only talking about, an amateur scratching the surface of the scientific evidence. This is all out there available. Stanley Miller was the one that encouraged all of us to say, well, since we're not doing so well in the laboratory, maybe we'll find, that there has been a chemical development of life in outer space. And so in 1974, he stated, we are confident that the basic process of chemical evolution is correct. So confident that it seems inevitable that a similar process has taken place on many other planets in the solar system. And on the evening of the first Mars landing in 1976, NASA's chief biologist, Harold Klein, asserted that if their theories of origins were correct, corroborative evidence for them would be found on Mars. Here's science at its best. Making a prediction, finding if the prediction is true. Guess what? No life on Mars. Likewise, in the Voyager 1 flybys of Jupiter and Saturn, no evidence to suggest life-generating conditions. And then people turned attention to one of the moons of Saturn, Titan, which might be more hospitable to life. But then it was found that the atmosphere of Titan was cold and dead. Indeed, it had a terrible atmosphere. 85% nitrogen, nearly 15% argon, less than 1% methane.